This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 393, recorded on June 10th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,400 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month. And for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
How have you been? Haven't heard it from you in 2 weeks or so.
I know,
right?
It seems really long time.
Yeah, it's good to be back.
Yeah, things are great. And it's 79 degrees Fahrenheit here, feels like 79, or Celsius, it's around 24, 25.
Have you had a little cold spell recently?
We did. It got a little cooler, like Tuesday when I got back from study section. Tuesday night and Wednesday, it was a little cooler, but it's been warming up ever since.
Yeah, we had a fierce storm go through in the last few days and brought a cold front. It's been in the mid-teens, 15, 16 during the day.
Wow.
It's just gotten up to 23 today. It is beautiful. No clouds in the sky. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And mid-teens, you mean Celsius?
Of course.
Yes. It's today, we had kind of the same thing here. Got all blustery. And,
Yeah.
today, it is frickin' gorgeous.
What's the temperature?
Just beautiful. It's 73 Fahrenheit. It's sunny. It's just light breezes. Beautiful, beautiful early summer day.
Also joining us from North Central Florida, Rich Condit.
Hey, hi everybody. How you doing?
Hey, what's the weather like there? Is it warm?
Oh, wow. Geez, you really caught me. Yes, it's warm. It's, according to my thermometer, it's 90 degrees, and I'm looking out my window, and it's basically a hot, mostly cloudy.
It's probably muggy down there, right?
It's pretty muggy. This says, my little thingy says it's 49% humidity, which doesn't sound like much, but that's relative humidity. You get up to 90 degrees, and 49% is humid. You know?
That's pretty sticky.
Yeah,
Yep,
it's not
pretty
humid.
sticky.
It's not humid here at all. It's quite dry because of this cold front that went through. All right, I want to tell you a few things before we move on to some science. First thing, as you know, we have been doing some advertisements here on TWiV and our other shows, and we're going to do more. We're trying to raise money so that we can all travel. I can get some production help, etc. I've also set up a place where you can go to give money if you'd like. You can go to microbe.tv/contribute, and there you can find a link to our Patreon site where you can subscribe. You can become a patron like the Medici, except you don't have to give millions of euros or lira, whatever they used back then.
You don't have to poison your relatives when they cross you.
You can give $1 a month, and we have a goal of $5,000 a month. And if half of our listeners, a quarter of our listeners gave $1 a month, we could reach that goal. And what would we do? Well, we could pay Alan to fly all over the place. We can travel more. So if I get invited somewhere, I can always say, "Hey, you guys want to come? We can pay for you." I can hire. I'd like to hire a production assistant to help do some of the work here and so forth.
That would be great.
So, and if you don't want to do Patreon, I also have a button where you can use your PayPal or credit card to give us a bolus of money. You could figure $1 a month for a year, $12 a year. You could give us $60 for 5, whatever you want.
Well,
It's all cool.
A regular contribution would be more of a drip of money than a bolus.
A bolus, yeah, that's right. Anyway, check that out. Also at Microbe TV, we have a new addition. Audiomunity has joined us.
Cool.
So that's
You did it.
That's cool. Yeah, it's there, and they because way back when I founded Microbe TV, I said, anybody doing science shows, join us. So Kevin asked to join us, so he's there. Also, Jesse, who does Bacteriophiles, is going to be on soon. I have to put him up in the next couple of weeks or so. And anybody else, if you're doing a science podcast, I think Nina Martin does one, right? I did ask her to join us. I haven't heard from her.
Cool.
But come and make us the clearinghouse for science podcasts. And then, of course, our next two shows are going to be on the road. The next one is at ASM Microbe, the meeting in Boston, Saturday, June 18th at 8 a.m.
Next weekend.
Yeah, don't forget, Alan.
No, I will not forget. It's on my calendar. I've got multiple alarms set.
You have to get up early.
Yeah. Oh, yeah. I'll be getting up very early in the morning to drive out there. But we have to be, the show's at 8?
Yeah, I think it's 8 or 8:15 start, but you should...
I'll be there earlier.
It says 8 o'clock here.
8 o'clock start. Yeah, you should be there a few minutes before.
Well, I should be there early enough to park and walk into the venue and all that. So
save your parking receipt. The Microbe TV will reimburse you.
Oh, cool.
And then the next one will be from ASV, Monday, June 20th. I think that's at 12:15, right, Kathy?
Correct.
And Kathy and Rich will be there. That's in at Virginia Tech in Blacksburg, Virginia. And by the way, our guest will be the keynote speaker. What's his name?
Excellent.
Stephen, it's in the...
it's in the ad, yeah.
In the ad.
I'm trying to find it, but there's too many emails.
Too many things we
Can't
have.
find them. Stephen Russell.
That's right. Yeah.
And I am assured by Grant McFadden that he's got a good sense of humor, because you couldn't beat Joan Steitz's sense of humor. Hers was great. And he knows a lot of things, and he'll be fun. So we'll talk about viral oncotherapy. He said he knows the whole field. Good.
Cool. Who do we have for guests at ASM?
At ASM, we have Erica Ullman Sapphire, and Galit Alter. Erica, of course, is from the Scripps, and Galit is in Boston. She was suggested by Erica because they collaborate. So she does interesting work in viral immunology. Of course, Erica's a structural biologist, so that should be fun as well. No, they're not live streamed. They're just recorded. So if you're at any of these meetings, please come. Please come by. We know you have a lot of things to do, but come by and check it out. At least come by and say hello. We'd like to see you. We do like people who listen to our shows. Okay. And then after that, of course, I think, then I'm away for a couple of weeks. We do have let's see, this is the last one for actually until July. This is the last TWiV here in the TWiV studio until July 22nd, right? Yeah. And then for the rest of the summer, I'm here, not going anywhere. Okay. Now, a little bit of science. Well, a lot of science, actually.
Yes. A couple of snippets and a paper.
So I was looking through snippety things. We've had a lot of Zika lately. As you know, the last episode was all Zika, 4 experts. But I did see this paper and brought up, I just wanted to touch on this issue of sexual transmission. There's a paper. It's actually...
Have we talked about that before?
We have talked about it, yeah. And it's an interesting... It's just a short correspondence in
very short paper.
The Lancet, one page. It's titled Late Sexual Transmission of Zika Virus Related to Persistence in the Semen. And what got my attention was the first sentence, which said, is written, sexual transmission of Zika virus related to infected semen is now well established. And I didn't really agree with that. I think, as you've heard us talk about before on TWiV, I'm not convinced that it's sexually transmitted yet, but the reference for that is a paper in New England Journal, Evidence of Sexual Transmission of Zika Virus, which I think we talked about a long time ago because it was published online in April. So in this paper, they have a couple who went to Martinique. It doesn't say where they live, but they live somewhere in France.
They returned to France from their trip to Martinique.
That's right. They returned to France. Exactly. Okay. They live in France, which we are told doesn't have Aedes aegypti later on in this note. So 60 and 61 years old. They returned to France, and then the man develops a rash, joint pain, no fever.
Right. I think he actually started developing symptoms while they were still in March.
The 4th. Came back on the 7th of February, and this started developing on the 4th. And so it turns out he has anti-Zika IgM and anti-flavivirus IgG in his serum. And they did show anti-Zika neutralizing antibodies. Urine sample on day 53, semen sample obtained on day 67. These are after symptom onsets. Were both negative for Zika virus RNA by RT-PCR. Woman, 40 days after her return, presented with a rash and joint pain without fever. RT-PCR on her urine was positive for Zika RNA. Serological studies were positive for anti-Zika IgM and anti-Flavi IgG. So they both had Zika infection. Now, so she got infected, and they had sex once a week a couple of times after they got back, unprotected sex. So this paper is saying that he gave it to her, and it was probably in his semen for a long time. Of course, his semen was negative at day 67, and the urine was negative. So I don't know how they can conclude that. And in fact, they didn't look at saliva, and of course, we know that Zika can be in the saliva. So I don't see why this couldn't be transmitted via saliva.
And he's 61, she's 60, so she's not pregnant. That's not an issue here. It's
just we would like to know. Do you get sexual transmission? So in my view, this doesn't prove semen transmission.
No.
Which is the title, related to persistence in the semen. They didn't even find it in his semen.
They didn't even find it.
So I don't get this. I don't get this whatsoever. So this, I went and looked at the CDC website. Now, they have a lovely summary you can go there every minute if you'd like. I call it the USA Zika Sit Rep. And so far, we've had 691 travel-associated cases in the mainland United States. And the CDC says 11 of these are sexually transmitted. And that's about 1.6%, which is not insignificant if you have a lot of people infected, of course.
Sure.
But there's also, on I think that same page from the CDC, some things we still do not know about Zika and sex. And one of them is we don't know if Zika can be passed through saliva during kissing. So that's what we've been saying is is kissing sex or not?
Right.
So it's sexually transmitted. If you say it's sexually transmitted, does that include kissing? Or because I know that nobody's reported transmission of Zika by just kissing, but on the other hand, that's not a reportable event like sex is, right?
Yeah.
If you know what I mean.
Well, they essentially anything that is clearly not mosquito spread, they're looking at sexual transmission as a possible mechanism. So I think if it was spread easily through saliva, that probably would have shown up by now.
So I think saliva's tough, right?
Sure.
Go ahead, Rich. Sorry.
So there's actually, I'm previewing a letter that's going to come up here. I think, to me, one of the compelling things is that the cases that are being reported here are all male to female.
Mm-hmm.
Yes.
Okay. And there's one male also.
And if it were, and one male to male, and if it were saliva, so that implies semen. If it were saliva, you would see bidirectional transmission, I think.
Yeah, I agree. The problem is the N here is 11 in the U.S.
Sure.
Right.
The other thing that I find interesting, at least, is then looking back at this New England Journal paper that they cited as evidence for sexual transmission. In that particular case, and I think that was a case study, they looked at RNA in saliva and in semen, and in saliva, the concentration was like 10 to the 4th copies per mil, and in semen, depending on when they actually assayed it, it was between 3 times 10 to the 7th and 3 times 10 to the 8th. Mm-hmm. So you're talking 1,000 to 10,000 fold higher concentration in semen than in saliva. So that and the apparent, at least in terms of reporting, unidirectional transmission is, it's adding up.
Well, CDC admits that they don't know if it can be passed through saliva, so
right.
I think that's important. I just would like to see more cases, right?
Well, in the sense of figuring out what's going on. We wouldn't actually like to see more cases.
No, I didn't say that well. You're right, but I would like to see more to really establish
more data.
Yeah.
So in situations where it becomes established that an infection is sexually transmitted, the evidence is primarily epidemiologic. Is that correct?
I think that coupled with finding virus in the appropriate locations, right?
Right. Or also, lesions in the, if it's a disease that presents with lesions in the appropriate location.
And then, of course,
well.
you can also look at the genotype and say this person gave it to that person. In the case of HIV, that's easy to do because you can easily see the diversification from a single virus. I'm not sure if you could do that here or not, but certainly you would like to see in a number of cases viruses in the right places coincident with sexual transmission. All I'm saying is that I just want to rule out. Just make sure that it's not saliva in some of these cases. Now, I think you should be careful. I agree that there is a potential, but I'm just being a little cautious with the data, just as we were in the beginning when the microcephaly connection was there, right?
Right. And there are many good reasons to practice safe sex that go well beyond Zika virus.
Yeah, of course.
This French couple, I guess uncharacteristically, was not at the barricade, so to speak. But that is probably a good idea if you've had a Zika virus infection or anything that might be transmitted this way.
I think the other thing that we could point out is that none of these presumed sexually transmitted cases are cases where protected sex has been used, right?
Right.
So that's telling, I suppose.
Yes.
Just want more. I don't want more, but I want more, if you get it.
Yes, you want more data.
More data.
So importantly, just to reiterate from the CDC website, of the cases that have shown up in the U.S., first of all, they're all travel associated. There's been no...
Continental U.S.
Continental U.S. There's been no transmission in the continental U.S. From mosquitoes in the U.S. Or, for that matter, sexual transmission. And the sexually transmitted cases amount to just 1.6% of the total.
Yep, right. That's right.
So if it's a mechanism, it is by far not the predominant mechanism.
Absolutely.
No. And now that we're getting into summer and it's hot and humid in Florida, and you certainly have the right mosquitoes down there for this, I would expect some mosquito-borne infection to start occurring. I mean, we certainly have it in Puerto Rico.
Yeah, so that's interesting because I've been thinking about this, and yes, I would expect a couple, but personally, I'm no epidemiologist or anything, but if you look at dengue and chikungunya, they've really not gotten a foothold in the U.S.
Well, and because it's a developed country with people indoors all the time. And it's very hard for a mosquito to bite a Floridian because they're, in air-conditioned spaces.
Yeah, Laura Kramer pointed that out last week. She said there was an outbreak of dengue in Mexico right on the Texas border.
Yep.
And it did not spread to Texas. She said there's just cultural differences, like the houses are further apart in Texas than they are in Mexico. And that in 80s is, sensitive to that sort of thing. It stays in a house. It doesn't really go house to house as effect. So,
and everybody's got the windows closed.
That too.
Yeah, the image that I have, I think Alan brought to my mind some time ago discussing the same thing, that, it's hot in the evening and everybody's home. And if you're in the U.S., you go inside and close the doors. And if you're in Mexico, you might go outside and sit on the porch where it's cooler.
Yep. That's right. So you Rich, you're inside now in the air conditioning, right?
I am inside in the air conditioning, and actually, when it gets really stinky, I'm just going to leave. I'm going to go to Oregon.
Have you seen mosquitoes in Florida?
Yeah, we were discussing this just the other day. We don't have many mosquitoes, and I think it's because of the mosquito control.
You mean we in Gainesville?
We in Gainesville, but I was I was visiting my brother on Amelia Island, and he said the same thing. They don't have many mosquitoes there. It's just not much of a problem, and I think it's mostly because of the mosquito control. I always too think about, we should look this up. The TWiV, the first road TWiV that we did.
Yeah, that's right.
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 About mosquito control in South Florida. We had the mosquito control people.
Yeah, that's right.
Yeah. Alan, you Skyped into that.
I Skyped in as a big face on the screen, right?
Yeah, Florida Gulf Coast University. I will look that up because that was a terrific episode where they really described a lot of the problems with mosquito control and the mosquito control efforts.
Yeah, I should listen to that. It would be relevant right now, wouldn't it?
Absolutely.
Forgot about that.
Right, and also the fact that the mosquito control, even in Florida, is a very fragmented affair that's county by county, and some counties have very robust, active, like the folks who were with the Key West or the Florida Keys Mosquito Control District, was it? And they were pointing out that other parts of the state don't have that at all. And, some do, some don't, and it's all funded at the local level, so it's very patchwork.
Speaking of mosquito control, there were 2 articles recently in PLOS Neglected Tropical Diseases, and I wrote about them yesterday on the blog, all about how the Pan American Health Organization had eradicated Aedes aegypti from 18 countries by 1970 using incredible mosquito control methods. And DDT and house-to-house checking, and the U.S. would not join in with that. They didn't want to have anything to do with that, and eventually the PAHO, the whole effort broke down. They didn't have enough money and so forth, so the mosquitoes came back. But Peter Hotez, who wrote one of the articles, said, U.S., as a consequence, has no good mosquito control infrastructure because we never joined them and we didn't learn it, but we need to reinstate it because we have to do something.
Well, frankly, none of the other places that did that have any good mosquito control infrastructure left either. Because when the program got abandoned, it all just went away. It was a derivative of the don't laugh, but there was actually a malaria eradication effort, which starting in the 1950s that sprayed DDT all over the place, and then the mosquitoes became resistant and more resistant, and more insecticide had less and less effect, and then the whole thing came tumbling down.
Well, what's remarkable is that Brazil was one of those 18 countries that no Aedes aegypti. How about that?
Yeah.
Now, the other paper is written by Paul Reiter, and he talks about how the PAHO effort worked, and he says, we can't use DDT now, but there are novel insecticides which can be embedded in a polymer that's sun and rainproof, and he said we should try these, and they may be good to...
Well, and it's important to point out that the way that was supposed to be carried out was an effort that would be very effective against a mosquito like Aedes. It wasn't supposed to be spraying DDT willy-nilly all over the environment. It was individual house-to-house spraying the walls with a specific concentration. And DDT is very, notoriously persistent. So you'd spray the walls, and for 3 months, the walls inside the house would just be mosquito death traps.
Yeah.
And you'd wipe out all the Aedes because, of course, that's exactly where it's breeding. It's not really out and about anywhere else. So that type of an approach could be very, very effective with a mosquito like that.
Yeah,
but not DDT.
Not DDT, right. I've forgotten what I was going to say, but go check out the articles. They're pretty cool.
So the Florida Gulf Coast University episode was TWiV 111. Live at Florida Gulf Coast University, the TWiV crew meets with members of the Florida Keys Mosquito Control District at Florida Gulf Coast University to discuss dengue in Florida and how to control it.
I remember what I was going to say. Paul Reiter says, in these locales, you have people driving trucks and spraying, plumes. He said this is totally useless because
not the right approach.
The aerosols stay up for a couple of minutes, and then that's it. Once they collapse, that's the end of the effectiveness. And he said, plus it scares everybody because these people wear suits, and they're thinking, why do they have to wear suits and I'm standing
here with just breathing it? Yeah.
So there's better ways to do it now. It's very interesting.
So another one was TWiV 178, our interview with Tyler Sharp.
Yeah, that's
Sharp on how tequila mosquito.
Yes.
One of the great all-time titles. And we meet with Tyler Sharp to discuss the Epidemic Intelligence Service and controlling dengue. And he had a lot to say about mosquito control.
And that's extremely relevant because he was, and I believe still is, in Puerto Rico, which is one.
He is in Puerto Rico, yes.
Yeah.
Dealing with this big time.
Big time Zika in Puerto Rico. That's right. All right.
Yeah, as a matter of fact, this is on your CDC website. Puerto Rico is looking at 1,259 locally acquired cases.
Wow.
It's exactly what Puerto Rico did not need right now.
Yeah.
Yeah. Or Brazil either for that.
Or Brazil either for that. Brazil about to host the World Cup and Puerto Rico filing for bankruptcy. This
Olympics.
is just...
You mean the Olympics, right?
Yeah, the Olympics, sorry.
They already did the World Cup.
The World Cup, right.
Yeah. Okay. We have a paper, and I have to tell you, last week, Charlie Rice was one of my guests at ASM Zika TWiV, and while reading his papers, I came across this one.
Yes,
sir.
Before we go to that, we should probably mention the Pittsburgh Zika virus lab
yes,
accident.
please do. Please, Kathy, you found that. Go ahead.
Kathy.
Our safety officer sent it to us, but it's been all over Twitter. So there's a accidental needle stick by a Zika virus worker in Pittsburgh, and they haven't given any details of where in Pittsburgh that is.
Got confirmation from Carolyn Coyne on Twitter that it wasn't them.
That it's not them, but there are other
Zika labs in Pittsburgh, yes.
And the person, so they know when she injected herself accidentally and when the symptoms appeared, and then I think she stayed home for a while. I don't remember that part, but then coming back now, she's not infectious to other people because the virus is, of course, transmitted by mosquitoes, and she's wearing long sleeves and long pants and insect repellent to protect the Pennsylvania mosquitoes.
And hopefully some needle repellent as well.
Yeah, right. So, but these are, one of the most common lab accidents when you're using needles. So,
Yep. Happens.
And it's not like there's a way to not use needles for a lot of things. So
if you're doing animal work.
Yeah.
Yeah, it's happened over the years many, many times. Fortunately, she is not pregnant, I presume, and it is not a serious infection in the non-pregnant, so that's good. Did they say that she have a rash? Yes, you said that, right?
She did have a rash, as I recall, yeah. Okay. It's already since last night that I read it. It's a long time ago. Too many things since.
Yeah. All right. So this paper I discovered while researching Charlie Rice, it's a letter to Nature. SEC 14L2 enables pan-genotype hepatitis C virus replication in cell culture. And the authors are Mohsen Saeed, Ursula Andreo, Hyo-Young Chung, Christine Espiritu, Andrea Branch, Jose Silva, and Charles Rice from the Rockefeller University and Mount Sinai here in New York City. And this is all about the fact that it's really hard to grow hepatitis C virus. By the way, another member of the TWiVerati. We got 2 TWiVerati today. It's very hard to grow it in cell culture. The virus discovered in 1989 took, I think, 11 years before it was able to be grown in culture. And it was only because this unusual isolate called JFH1 could replicate in hepatoma cells in culture. And that was from a case of fulminant hepatitis, so it's an unusual case. And that's, of course, been great to study infection, but there are many, many other isolates, and it's very hard to propagate them in cells without selecting mutations in their genomes that adapt them to cell culture. So they said, well, there must be something missing in cells that's compensated for by these adaptive mutations. So let's look for it. Yeah. Let's do a screen to look for what in a cell is either inhibiting replication or is missing and is needed for replication. So they do a lovely screen to answer both questions. They do one where they will take cells and they put a library of short hairpin RNAs, which would knock out genes and mRNAs in the host cell and look for an inhibitor, or they add a cDNA library in and ask, is there something we can add back and make the virus replicate? So the way they do the assay is really cool. They have a selectable marker, a drug resistance gene in the viral genome.
Which has been around for a long time. That's their Replicon assay. That was one of the first assays they came up with for any kind of replication of hepatitis C in culture, where you just you remove the structural proteins and substitute in a drug selectable marker. and transfect, I guess, an RNA transcript from that thing into cells. And it is, under selection, maintained as, they call it a replicon, but it's essentially a persistent infection with this replicon.
They're not making virus particles. It's just RNA replication. And in this case, they're assaying it because they have a drug resistance marker. They apply drugs to the cells, and then you get colonies if the genome is replicating.
Right.
And they use...
Now, you would normally, in order to get that genome replicating, you would need some compensation for whatever the normal block is, which they didn't know at this point.
So that you'd have to have what they call adaptive mutations in the replicon, right? But for this assay, they use a replicon that doesn't have these.
Doesn't have the adaptive mutations.
Yeah, they said. And so the shRNA library didn't give them any results. So there's nothing replicating if you take away genes. But the cDNA library gave them a lot of colonies. And they do a cool experiment. They treat these colonies with an anti-HCV compound to get rid of the replicon, and then they reintroduced the replicon to show that the cell's been altered and not the
That
virus.
part was amazing to me, that you could that you could do that.
Yeah, it's very cool.
2 drugs that they give. I can't find it at the moment, but yeah.
Yeah, it's really cool.
So they go in by putting in this replicon that's the virus that shows whether or not the virus can replicate in there without these normal modifications, plus any of a random set of genes from the human cDNA library. And they found the combinations that way that worked, and then they removed the replicon, put it back in, showed that it still works, and then assayed it the other way and made sure that they'd gotten the right answer. Very, very cool.
Yeah,
and I found the 2 drugs. They're declatosphere and 2-CMEA. 2'-CMEA. Anyway, they're in extended data figure 1. That's why.
A lot of extended data in this figure.
Yeah.
Yeah, and it's a letter.
It's a letter that's like 5 pages of text and then like 10 pages of data. Yeah.
So what's in these cells that allows the replicon to replicate? They did sequencing of the cDNA that went into these cells from 45 colonies, and they all had the same gene.
Yeah, that's pretty telling.
That must have been a great day.
Yeah.
I mean, this is what you dream about, this kind of result. Yeah. You say, I want to do this screen. No, it's not going to work.
You're going to pull out all kind of the junk. Yeah.
And they get the same thing. I mean, I would say you get 5 of one and then 1 each of many other things, right?
Yeah.
They got 45 of the same gene.
Right. Yeah. And by the way, the way this is done is you, in doing the screen, you transfect in this cDNA library, and each of the genes there were like 7,000 genes. So it's not everything, but it's a lot.
It was enough.
And each on the cDNAs, when you transfect them in, they have some unique constant sequence flanking the cDNA. So you got a set of primers you can go in afterwards and specifically amplify whatever cDNA went into the cells. And they got the same thing in each case.
And there are so many reasons why this might not work.
Yes.
Right. Like if this requires 2 genes for the virus to be able to replicate, you're probably not going to find it this way, because each cell is not going to get. You'd have to get exactly the right 2 genes in there. So yeah, they really they were skillful and lucky.
It's wonderful. There's so many screens like this that have not worked, which we never hear about. But this is just so beautiful. It's lovely. So this gene, SEC14L2. It's got a couple of names, but they're all, actually, I guess...
None of them are any catchier than that.
Supernatant protein factor 1, tocopherol-associated protein 1. Anyway, it's a cytosolic protein, and it's a member of a protein family that binds lipids, found in many tissues. They could not find the RNA or protein in human hepatoma and non-hepatoma cell lines, but primary human hepatocytes, both fetal and adult, made the protein.
Which is also... nice to see.
So they make a stable HUH. So these experiments, by the way, are done in a hepatoma cell line called HUH 7.5, which is known to support very well the replication of the replicon. They stably produce SEC14L2 in these cells, and then they put RNAs from a variety of wild-type replicons of all the genotypes, 1A, 1B, 2A, 3A, 4A, and 5A. And they do the G418 selection, and they get colonies for all of them. So this allows the replication of replicons of all the genotypes that they tried. It's very cool. Very good replication. They did it in a couple of different cell lines. So it's beautiful. They also do a cool set of experiments where they place the SEC14L2 under the control of an inducible promoter so that they can turn on and turn off the production of the protein. And they can also regulate the level of the protein by this system by varying the amount of the doxycycline is the inducible control here. And they show that the more SEC14L2 in the cell, the more colonies. And when you turn it off, the replication stops, so it needs to be around all the time.
That last experiment I like the best. When you turn it off,
yes.
So it's not a permanent alteration of the cells. It's got to be there all the time.
Yeah.
Now, if you do this assay with cells containing this SEC14L2, if you use cell culture adapted replicons, you don't get much of an increase with the protein, which is interesting. So it's telling you that the replicons have mutations which are somehow doing something that is related to what SEC14L2 is doing. Is
that true, or is it just that you can only do so good?
I don't know. I don't know if these the replicons. I'm not sure the replicons are so good. I mean, you can do better.
Right.
I don't know. Could be either one.
I spent a little time looking at what the adaptive mutations in the replicons were, because that interested me.
Yeah.
Mm-hmm.
And let me see, I noted it here somewhere. That actually they're in a lot of places. They're non-structural proteins, NS3, NS4B, NS5A, and NS5B. Now, NS5B is the RNA polymerase. The others are non-structural genes that mostly have functions that are implicated in the innate immune system. So it's as if maybe you can adapt by getting slightly better RNA replication, but also somehow thwarting innate immunity somehow. But I don't know how that relates to this SEC14L, but we can discuss that.
Kathy found a paper that I think is relevant, and once we talk about what this does, I think it'll make sense.
Okay.
So hang on.
Right.
So then so far this has all been done in Replicon, so now they say, does this protein have an effect on replication of full-length viral genomes? And so they have this lovely assay for infection. Turns out that HCV infection causes a mitochondrial protein to relocalize to the nucleus, so they can tag this mitochondrial protein with red fluorescent protein and watch it go in the nucleus.
Have we talked about this before?
Because we have. It sounds
very familiar.
Right?
Yeah.
I believe we have a long time ago. Yeah, It's a very cool assay. And so they show that this protein allows replication of full-length HCV in cells. And they also can insert a selectable marker into the full-length genome and show that this protein allows the establishment of colonies with full-length genomes in them. And they also do a cool thing. It's been hard, apparently, to take serum from patients with hep C and get virus to grow in culture. So they actually inoculate cells producing the SEC protein with patient sera, and they replicate.
This is another one that just can't believe that worked.
Because here, if you take serum and put them on HUH 7.5, you get crappy replication or nothing. And now with this protein, it's really, really cool. It's going to enable a lot of things. They start to ask about this protein. It's got a couple of different domains, and they change each one of them, and each one interferes with this ability, so they can't localize the activity to a particular part of the protein. They don't find any interaction between SEC protein and the components of the hepatitis C virus replicase proteins. They look at a variety of signaling pathways. They look at cholesterol synthesis, no role for this protein in those. And then they turn to the role as a vitamin E binding protein, because I think it's known that this protein binds vitamin E.
Right.
And vitamin E, of course, is an anti-lipid oxidant, right? So typically in cells, lipids can be damaged by free radicals, and that messes up membranes. And in fact, just aside, while I was looking at this and following the link that I think Rich provided, the first organic free radical was identified at the University of Michigan in 1900 by Moses Gomberg. Go Blue.
Mm-hmm. That's what he's famous for.
Really? I put that in so you would be happy.
Yeah. And and he never married, and he donated he left his legacy to the chemistry department. So they have a lecture in his name, and
it's cool.
Other stuff. So yeah.
So lipid oxidation can happen, and by free radicals, it damages the lipids, damages the membrane. Vitamin E protects against it. It's an antioxidant, and apparently SEC14L2 enhances the accumulation of vitamin E in these cells that they're using for these assays. They have, and hep C is particularly sensitive to lipid peroxidation. And there are isolates that are resistant to lipid peroxidation, and these are not enhanced by SEC production, which enhances the vitamin E production.
Which I think wraps back around to Rich's question and Kathy's answer.
So
now Kathy can talk about that paper, which I think is relevant to this,
yes.
Right?
Right. So this is a paper from Stan Lemon's lab, and the first author on that paper... it's not clicking through for me. This came out in 2014. First author is Yamin or Yamani. And basically they show that the replicase, which Rich mentioned a moment ago, is the I'm off that page now.
NS5B.
NS5B. That's the polymerase part of it, but the complex also includes the NS3,4A protease to make up the replicase. Mm-hmm. And that when you have the normal replicase molecule, lipid peroxidation restricts HCV replication. But you might remember a minute ago, Vincent talked about that special JFH1 HCV strain that can grow in cell culture. Its replicase is not sensitive to lipid peroxidation. So that was a clue that, again, when you guys were saying that they got 45 clones that were the same thing, I'm sure that they knew about this paper from Stan Lemon's lab, and that their minds probably already went toward, maybe this is related to this lipid peroxidation thing.
Right.
And the implication is that somehow, where did I write that down? That the HCV sensing this lipid peroxidation, somehow its replication is reduced, and maybe that's what leads to persistent infections by HCV rather than an acute infection if the replication is damped down.
So do we have any mechanism for what's the connection between lipid peroxidation and restricting RNA replication?
Yeah, well, that's the key, right? I mean, I don't think we know, but, RNA replication does require membranous vesicle structures.
Okay.
So that may be involved in that.
The other thing that I noted is that the lipid peroxidation not only screws up the lipids, but downstream from that, when these things decompose, there are some toxic byproducts of the decomposition that are specific to the lipid peroxidation. So it's not necessarily just about the lipid.
All right. Lipid peroxidation. So they do a couple more experiments to cement this idea that SEC14L2 is somehow involved in reducing the effects of lipid peroxidation. So if they grow the virus in medium with serum that's delipidated, depleted of lipid-soluble materials, including delipidated fetal bovine serum, depleted of lipid-soluble materials, including vitamin E. This protein doesn't support replication in those cells. And the other thing is one of the products of lipid oxidation is malondialdehyde, a secondary product of peroxidation, and they show that SEC14L2 reduces the abundance of that product, which is consistent with the idea that it's inhibiting lipid peroxidation. So the idea is that this protein is it's enhancing vitamin E mediated inhibition of lipid peroxidation.
Vitamin E by itself won't do it.
So there's something else, right? Additional functions as well. Vitamin E doesn't do it, but somehow SEC is working through vitamin E.
Well, or Sec14L2 binds vitamin E or something along those lines. It assists vitamin E.
They didn't look at whether it binds, right? They just show that it stimulates it increases the concentration of vitamin E in cells, right?
Yeah, but I think they if I'm not mistaken, they cited evidence from elsewhere that it actually binds vitamin E.
Right.
Yes. Yes.
Okay. The antioxidant effect of SEC14 is mediated in part through VE, vitamin E. Yeah.
Right. They talk about the role of Sec14L2 as a vitamin E binding protein.
Yeah.
Give a couple of references for that. So, yeah.
So I think this is a cool paper, not only because they identify a protein, but they start to figure out how it's assisting hepatitis C virus replication. And it's also a great tool. You don't need adaptive mutations anymore, which is nice. You would like to study the wild-type genomes in these cells.
And apparently you can just pull out clinical isolates now and put them on these HUH 7.5 cells with the Sec14L2 and go to town.
Yeah, that's a very important...
that's awesome.
Breakthrough.
This journey to get HCV to replicate in culture. I think Charlie said on the podcast you did last week, that it's been 15 years.
Yeah.
And it's, there are... For the uninitiated, there are many examples out there of viruses that won't grow in cell culture. And people try all kinds of stuff to get it to work.
People try stuff exactly like what's in this paper.
And it's so difficult and so frustrating. And I think that this whole effort from start to finish over the whole 15-year period has been a remarkable evolution of figuring out how to grow a virus in culture. It's just amazing.
Yeah, and of course, Zika, there's no issue. It grows.
Right. There are other viruses, exactly, that just...
I wonder if, when Hep C was first discovered, it was many years ago, 1989, I wonder if there would have been more people working on it immediately if we could grow it in cell culture.
I'll bet,
right?
Absolutely.
Yeah.
Sure.
Because here, Zika, you can grow, and it's BSL2, and so people are moving to it, and they didn't do that with Hep C. I mean, a lot of people did. Charlie was one of the people who moved to it immediately, right? And that has really enhanced his career. And he moved to, his whole lab at Rock was financed in large part by the Greenbergs, whose son had Hep C. Right? So it was a big deal for him. It's pretty cool. Anyway,
another example of a virus that doesn't grow in culture is human papillomaviruses. You can create a fancy, and that's probably never going to happen because of the biology, which we could go into another time, or we have to in the past. But you can create a really fancy thing called a raft culture and make it happen. But
and there's a couple other methods too that you can use.
But there again, if you'd have been able to grow it in culture, it probably would have advanced much more rapidly.
Yeah, absolutely. I'm sure that it's not as simple for HPV as one gene like this, right? It's definitely not as simple.
No.
This episode is brought to you by CuriosityStream, the world's first ad-free nonfiction streaming service founded by John Hendricks, who founded Discovery Communications. They have over 1,400 titles and 600 hours of content available in 196 countries on many platforms, not just the web, but Roku, Android, iOS, Chromecast, Amazon Fire, and Kindle, and Apple TV. And I think the reason why we're pitching this to our listeners is because they have a wide variety of science and technology content. It's all nonfiction. They also have nature, history, and many other topics as well. They have a lot of 4K content, which is very high definition. And they also have interviews and lectures. They have Stephen Hawking's Universe, Next World featuring Michio Kaku. They have a few programs on viruses, including one on Zika. A Life on Us, a series exploring the biodiversity of our bodies, and much, much more. With Curiosity Stream, you get real science shows, not just reality TV science shows that are on cable TV. There are monthly and annual plans, and they start at just $2.99 a month, less than a cup of coffee or the cost of one title on competing on-demand platforms. Check out curiositystream.com/microbe. Use the promo code microbe during sign-up to get unlimited access to the world's top documentaries and non-fiction series completely free for the first 60 days. That's 2 entire months free of one of the largest 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign-up. We thank Curiosity Stream for their support. I keep wanting to say startup. I don't know why. I've done it several times.
Well, we thank them for starting up, too.
Too. I also want to tell you about the annual meeting of the American Society for Virology. It's being held this year. And in fact, it's a week from tomorrow it's starting, or today, actually, the pre-meeting meeting. Do they start?
A week from tomorrow.
Tomorrow, Saturday. Blacksburg, Virginia, on the campus of Virginia Tech, June 18th through the 22nd. Of course, if you're just hearing about it here on TWiV, it's too late probably. You could still go and register there, but you're probably already going. But we'll tell you anyway about the local organizers, XJ Mang and Zach Edelman. Their committee arranging this venue, the keynote address and plenary symposia arranged by ASV President Grant McFadden. This year's keynote will be from Dr. Stephen Russell on oncolytic viral therapy, followed by 5 days of symposia workshops, poster sessions, and state-of-the-art lectures. Come listen to Dr. Joe Handelsman talk about leading the White House Office of Science and Technology Policy. Be part of a live TWiV audience and enhance your presentation skills at the Career Development Workshop presented by Jean-Luc Dumont. Also this year, ASV is hosting 2 late-breaking Zika virus oral workshops and a poster session. You can still register on site. Go to asv2016.cpe.vt.edu or asv.org for more information. Hope to see you all there. All right, we have a lot of email.
Yeah, the backlog is kind of getting big here.
Oh, and this is just the tip of the proverbial iceberg.
Oh,
there's so many
maybe
more.
We should do an all-email episode.
Bingo. Yeah, we should do one, except we don't have one until
July sometime.
July.
Right.
Hopefully I can get Ron Vale back at that point. But yes, we'll try and make a little dent. We have only been talking for 1 hour, so we can do a half hour of email.
All right.
And I have grouped a bunch of Zika-related emails together. And the first one is from Zach, who writes, "Hey, TWiV team, reporting from Woodbridge, Virginia. The weather is currently 64F, 4 degrees Celsius, humidity at 50%. Is that possible?
That doesn't sound right.
4 degrees is just a few degrees above freezing. That can't be right.
That can't be right.
How long ago did this email come in?
No, it's not that long ago.
Right, but it's either 64F or 4C is correct. I don't think they could both be.
April 7th. Not that long ago.
Okay.
I am somewhat of a recent listener. I started in January 2016 and have been working my way through some of the backlog of episodes. I really enjoy the show. I am currently taking an infectious diseases course focusing on viruses, and this show has been very insightful. I appreciate the knowledge and perspective of all the team members, including guests. I recently had to present on Zika virus and found this podcast helpful in stirring ideas. I was curious on everyone's thought. I came across an article talking about what was making Zika virus worse in the outbreak in the Americas than previously reported in Asia and Africa. Dr. Michael Diamond at Washington University and officials at WHO were theorizing that individuals with previous dengue virus infection were more susceptible to severe Zika virus. And is this theory more likely than possibly a lack of immunity of individuals in the Americas or possible mutations with Zika virus, considering many of these side effects were not reported before, whether not taken into account or simply not there? Thanks, Zach. Well, I think we just don't know. I think we're going to learn in the next months whether dengue antibodies are making disease worse or not. But I think it's not even clear before the Pacific outbreaks, there was not a lot of infections that were documented, so we really don't have any data to make a comparison. But certainly now, looking back at the Pacific outbreaks, there is clear evidence of Guillain-Barré and microcephaly. So I'm not so sure that there's a major difference. What do you think, folks?
Well, I know that at the Southeast Regional Virology Conference where Scott Weaver talked, I definitely got the impression that he thinks that that these kinds of things may have been there all along.
Yeah,
right.
That's the impression I got, and that maybe there's not even evidence for any kind of increase in transmissibility or anything like that. So,
right.
Yep.
Yeah, my sense is that until it hit South America, the populations that it was working on were just too small to see what was going on. And now all of a sudden it hit very dense populations with economic conditions that are ripe for the mosquitoes and et cetera.
Yeah.
And it just took off. And let's put it this way. There's no compelling evidence that either the genetic variation in the virus or the conditions such as the antibody-dependent enhancement have anything to do with this.
Right.
There are things to think about, but there's no compelling evidence, I don't think, at this point that they're factors.
Right.
Yes. In order to argue that there's been a change, you have to have some kind of historical data. And in this case, we have an almost perfect void of data in the historical record regarding Zika. It's a case here, a case there, poorly documented, and just nobody had any clue what was going on, and this was not even really studied until it hit a place that had enough population that it got some attention.
right. I'm trying to remember what we think is the current percentage of incidence of females getting pregnant and getting microcephaly. Is it around? We think it's around from 1 to 13. Is that the range?
Yeah, that's
from
exactly
1 to
right.
13 is the range. That's right.
Right.
Yeah, that's exactly
So, and then just thinking back what we were saying about the prevalence of sexually transmitted around 1.6%.
Mm-hmm.
So you can imagine that if there's just not that many cases happening in Africa or elsewhere, that those kinds of things are not going to appear above a background. And
Right.
Mm-hmm.
Or if it is happening and nobody's noticing.
Yeah.
Right.
I mean, if it's happening in rural areas where there's a background of a huge number of other health problems, and nobody's keeping track because there's no health infrastructure, it's going to be totally silent. This stuff could have been going on all along.
Mm-hmm.
Yep. This is actually related to the next email from Paul, who wonders about this New York Times article saying that the virus mutated from an African to a pandemic strain a decade or more ago, then spread east across the Pacific from Micronesia and French Polynesia till it struck Brazil. While the merry TWiVsters say there is no evidence at all for such a theory, could this be another example of journalistic catastrophizing? But the problem is, Paul, that this article was written by a scientist.
Yes.
It was written by Peter Hotez, and he says the most plausible theory is that it mutated and became pandemic. And there's just, as we have said, there's no evidence for that whatsoever.
No.
And at the last week, just before the Zika TWiV, there was a press conference with a number of flavivirologists, including Michael Diamond, and he was asked this by one of the journalists, and he said, there has been no systematic comparison of different strains with respect to transmission or disease. That's a quote from Michael Diamond. So,
Doesn't mean it's not being done.
oh, yes, I'm sure it is being
Well,
done.
the two papers that we did a couple of weeks ago, they had every opportunity.
Yeah, that's right.
That one of them was the transplacental transmission of Zika, and they had both strains in the paper, but
Yeah,
they didn't do
it.
the experiment with both strains, and I'm really frustrated by that.
Yep.
I'm of course, totally laid back.
Of course, no.
But I get. I'm focused on that one.
This is a very difficult question to answer in people, of course,
Mhm.
right? You can do a lot
Absolutely.
of cell culture and animal experiments, but to do it in people is extremely difficult. You may find differences in other models, but correlating it to people is hard. But yes, as Kathy has said, people are working on this all over the place. It's a big issue, and we'll see. We'll talk about it here when it gets published.
But I do wish that
or preprint published.
People, especially, like Peter Hotez, who is on a lot of these sorts of things, would stop theorizing beyond the data, and this is going to get planted as the notion of what's going on.
Yes, I agree. We just
don't have the data to say that.
This time we cannot blame it on the journalist.
No,
we can blame it on a scientist. Actually, I should point out that's an opinion piece in the Times.
Yes.
Right. Mm-hmm. Okay, Kathy, can you take the next one?
Oops.
You want to pass?
No. Sean writes, "Good afternoon, professors. My good friend, a fellow public health graduate student, shared this MMWR article with me about male-to-male transmission of Zika virus in Dallas. I remember from the past few TWiV episodes featuring this en vogue virus that it was still unclear whether or not Zika was transmitted sexually. Perhaps this article will garner more attention from other reproductive health enthusiasts such as myself." He gives a link to the MMWR paper. "It's a lovely 25 degrees Celsius, 77 Fahrenheit with blue skies here in LA. Wishing you all a warm spring day and kindly requesting any melted snow to be sent to our mountain so we may continue to recuperate from our drought. Mm-hmm. Sincerely, Sean." And then we got a link from this to the same MMWR article from Ted, who writes, "Probably the first of many reports to follow in this population, and adding to the Zika story. Continued thanks to you regulars and guests for the always informative podcasts." So this MMWR, that's the one that
he's mentioned before.
We had mentioned it before, right?
Yep.
So we're just catching up.
All right. Alan, could you take the next one, please?
Sure. Stephen writes, "Dear TWiVerati, I have to say that while I was initially skeptical about Zika sexual transmission, I've been won over, and I'm surprised that you're still skeptical. Caveat 1, I'm a layman, although TWiV and other sources keep me informed. Caveat 2, I had the pleasure recently of hearing Dr. Brian Foy talk about sexual transmission of Zika. One thing that I haven't noticed you cover yet is the sex disparity of non-vector transmission. These cases are from males to females, and in this week's MMWR, from male to another male. If the non-vector transmission were from something else, say exposure to rashes, one would expect a two-way transmission, but having it be just from males, or even if the preponderance of transmission was from males, makes sexual transmission the easiest explanation. Yes, see this episode above. One thing Dr. Foy pointed out that makes him think sexual transmission is common is that in earlier outbreaks, there's a noticeable sex disparity in some of the earlier outbreaks where more women than men were infected. One other comment, more minor correction, though it has some bearing on the debate. I heard Alan say in one episode that the cases of alleged sexual transmission were all apparently transmitted during or shortly post the symptomatic period. From Dr. Foy's original paper, quote, "Furthermore, patients 1 and 3 reported having vaginal sexual intercourse in the days after patient 1 returned home, but before the onset of his clinical illness," end quote. It's warm now and right now in Fort Collins, but we're supposed to get snow over the weekend. Apparently this was a letter from April 15th. Thanks for all your hard work, Stephen.
Mm-hmm. I think we talked about this already.
Yeah. I think it's still quite a stretch to say that sexual transmission of Zika virus is common.
Right.
Yeah.
1.6% from the U.S. data, I would not call that common at all. And the sex disparity is actually pretty easy to explain, considering that these are mosquitoes that are found most associated with dwellings.
Yeah, I was thinking about the mosquito explaining part of that.
And in especially in poor rural areas, who's at home?
The women.
Oh, interesting. Okay.
So I wouldn't hang too much on the sex disparity of it, because the women and men are going to be in different roles.
Isn't Brian Foy the fellow who actually transmitted it in Fort Collins to his wife, right?
Mm-hmm.
Yeah.
Yes.
Yeah.
It's common in his household, yes.
Rich, take the next one, please.
Steen. Is this Steen? Steen writes, "Dear Vincent and friends, thanks for reading my message and discussing the cowpea mosaic virus paper, TWiV 380. I love hearing about the history of structural biology. For what it's worth, Hull writes that of 92 plant virus genera, 36 have multipartite genomes, and that many of these include multi-component viruses, genome segments packaged separately." This has been in the queue for a while. This is back from the time when we discussed a couple of these multi-component viruses, and we were amazed. Hull lists multipartite genome structure as one of at least 12 strategies for increasing accessibility to host translation machinery. I really don't need a textbook. I guess we offered him one.
Yep.
I borrow an old edition from the library sometimes, but I look forward to future contests. I'm planning to graduate and move away soonish. I need to avoid accumulating more heavy books. Regarding Jared's questions, I poked around a little bit, but did not find much about Orchid Symbiont viruses.
Yeah, someone wanted us to talk about them.
Mm-hmm.
Well, if Steen can't find anything, well, he's the plant virologist.
Yeah.
Yeah, we were wondering if this multi-component was special because of the way that viruses are transmitted in plants. Obviously, it's not the only way. Obviously, there are other kinds of viral genome structures as well.
Yeah, so, what blows my mind about them is the multi-component viruses. You got to deliver at least 2 particles, depending on how many components there are, to a cell simultaneously. Yeah. But the delivery mechanisms may cover that.
Do either of you know that why the multipartite genome increases accessibility to translational machinery?
Yeah, it's just if each one encodes an open reading frame, right? That's just another it doesn't really increase it. It's just another strategy for getting multiple proteins from a genome. You break it up, right?
It's not quite how I read what he wrote, but okay.
Yeah, I didn't see how it would increase the accessibility.
Right.
You've got a certain number of ribosomes around, and that's that.
The next one's from Ricardo. Hello, TWiV friends. A phage, in order to infect a bacteria, needs a key to open its access. What if that key gave access to multiple misfolded proteins such as amyloid beta and led to its destruction? Viruses can be our friends. And he sent a link to a PBS Nova article, and I went and looked at the some of the original literature here. This is really a cool story. It turns out that starting from some experiments in mouse models of Alzheimer's, a control experiment revealed that M13 bacteriophage by itself, which was a control for delivering an antibody to these animals, actually on its own reduced the amount of amyloid plaque in these mice. And it turns out that there is now a whole field of treating various amyloidoses like Alzheimer's and Parkinson's with phage. There's studies going on. I hadn't known this.
Cool.
This is very cool. And there's a company in Boston doing this. There are lots of papers in the literature. So I'm wondering if at some point we should do an original paper on this.
That would be good. The PBS article, this letter's been in the queue for quite a while as well.
Yep.
And it's confusing enough so that every time I have to reread the article. So
Me too.
There's a couple of really cool things. First, you mentioned it, Vincent, but the serendipity that brought this up is great because there was a researcher who was trying to treat Alzheimer's in a mouse model with an antibody, and to deliver the antibody, fused it to M13, which is a common technique, and as a control had just M13 alone, and it worked, but M13 worked as well as the antibody, and it was her son. Is that right, Kathy?
Yes. Who
ultimately picked up and decided to develop and ultimately commercialize this further. And it turns out that it's the virus attachment protein. The M13 is a filamentous single-stranded circular bacteriophage. And at the end of the filament, there's, I believe, a single copy or a few copies of a protein that's required for attachment. And this protein has the property that it can effectively melt or disaggregate aggregate amyloid aggregates. And I presume that that's part of its mechanism for attaching and penetrating the E. coli. And they've identified the active domain from this and now made a fusion with an antibody for the purposes of delivery, so that now they have an antibody where the active site of the antibody is this amyloid disaggregation domain from M13, and it disaggregates amyloid plaque. It's incredible. What an amazing
That's
story.
amazing.
Yep. All right, now we don't have to do the paper. You just did it.
Yeah, we got to do it.
Explained it well. It's amazing. It's just amazing. And if it turns out that this is useful clinically, wow.
Yeah.
I guess there's a few copies of this protein.
Yeah.
Yeah. It looks
But the story, too, about how this woman's son developed this thing over time is really cool.
Yeah.
So that article is, it's full of human interest, right? Because that's what makes it appealing. But if you want to just get to the story, it takes a long time to get through to actually what happened.
But the actual, one of the interesting things about this is that this protein works on, as you said, a number of different of these aggregates that are associated with various pathologies. So if you could really develop this, it could have quite an impact on healthcare.
Yes.
Sure. Yeah, in fact, I put a link to a review paper, which is called Phage M13 for the treatment of Alzheimer and Parkinson's disease. And the author is Messing, who developed M13 years ago, right, as a vector, particularly for sequencing, which is what it was used for subsequently. But he's been big in the biology of M13 for years, and now he's talking about it.
Have you guys worked with M13?
I have, years ago, yes.
Because that's how we did all the shotgun sequencing in the D frame. I loved working with M13. Great phage.
Yeah, lovely plaques.
Great
Grows fast, easy. Yeah.
Yep. All right, Kathy, you're next.
I'm still. Is this Helen now?
Yeah, it's Helen.
Okay, good. Helen writes, Dear TWiVome, I'm a PhD student at the Novo Nordisk Foundation Center for Protein Research at the University of Copenhagen, and my work focuses on bioinformatics methods for inferring virus-host protein-protein interactions. I discovered TWiV after finding Vincent's virology courses on YouTube, started listening, and now I look forward to new episodes. The lectures and TWiV have been invaluable starting points for me to locate background material on virology and to keep up to date with advances in the field. Thank you for your outreach. Vincent mentioned in passing in TWiV 380 that there was some evidence that virus infection was beneficial for its natural host. I'm unable to find more details about this beyond those that apply broadly to all DNA viruses, even after checking page 2 of Google and previous TWiV episodes tagged with symbiosis. Could you point me to a reference? I would be very interested to read more. Best regards, Helen.
What I meant to say is that if the virophages inhibit lysis induced by viruses, and that's how they protect the host. So it's not correct to say they're beneficial. It's kind of indirect. But there are a couple of articles that we can show you about that. Yeah. Alan, you're next.
Yes. Anthony writes, this link is on climate change denial, but it struck me that the anti-vax sentiment is something that can be investigated via psychoanalysis, too. Some months back, a Google search showed no results. The Institute for Psychoanalysis in London, England, but the APA, American Psychoanalytic Association, is another link. Perhaps it might be suggested to the APA that the anti-vax belief might be an interesting topic for a psychoanalyst to research, and sends this psychoanalysis of the climate change deniers movement.
Do you remember our discussion with Seth Mnookin?
Yes.
He talked about this a bit, right?
Yeah, and I think we've kind of touched on this a few times in the past, the psychology behind these counterfactual belief systems and people forming, identities around it and becoming entrenched despite the facts.
Alan, your wife is a psychiatrist. Does she have any particular thoughts about anti-vaxxers from a professional viewpoint?
No, not particularly.
All right.
She finds them highly annoying, but
yes, as we do as well. Yes, of course. All right, Rich Condit.
Bridget writes, Hello, Doctors Racaniello, Despommier, Dove, Condit, and Spindler. It is 37 degrees Fahrenheit, 3°C, and overcast while I'm writing from Columbus, Ohio. We're getting further back in the queue here. I am a relatively new listener to the program, but I'm hooked already. I value your insightful dialogue and in-depth discussions about current research in virology. Since graduating from undergrad at Ohio State last summer, my life has been relatively void of conversations involving molecular biology of any sort. I missed those discussions enough to turn to YouTube as a learning tool, where I discovered Dr. Racaniello's introductory virology class. Within 2 months, I had watched all of the 2014 lecture videos and had begun listening to TWiV. Prior to watching those lectures, I knew that I wanted to pursue biomedical research, but was struggling to identify a specific area that I was passionate about. That changed quickly. After each new lecture, I found myself wondering a million things, most of which haven't been answered yet. Because of my incessant curiosity, I am now quite convinced that I want to study virology in graduate school and beyond. In researching various PhD programs, I've become a bit overwhelmed, in a good way, about the wealth of viral research occurring in the world. I am writing to you all for some advice. Currently, I'm extremely interested in the various oncolytic strains being developed. However, some researchers I have spoken to have given me the impression that this area of research has many drawbacks and may not be a wise choice of research topics. I tend to enjoy more molecular biological-based research methods to biochemical assays, and I greatly appreciate research that goes towards understanding basic molecular biological systems rather than specific outcome research. Applause! Applause!
Yes.
i.e. antiretroviral drug development, and I am pretty flexible there. Are there any ideas of research that you all are particularly interested in or seem promising? Are there any areas that seem to be less viable? Who are some up-and-coming researchers in the field that would be worth keeping an eye on? Do you have any advice for choosing a PhD/focus program? Sorry for such a long message. Thank you for taking the time to read it and think about these questions. Also, thank you for your slightly skeptical analysis of each paper you present. That's critical thinking. Since I began listening, I find myself more willing to trust my instincts when something I read in a journal article seems overblown or speculative. The ability to evaluate claims is fundamental to research. Your weekly discussion gives me a model for what that process looks like among practicing scientists. As someone who is just starting out, that is extremely valuable to me, so thank you. Best of luck in all your future research, Bridget. What a delightful letter. Holy cow. She has wrung out of our stuff exactly what we want people to get. Fundamental research, critical thinking. This is glorious.
And she wants to be a virologist.
So the first thing that comes to my mind, Kathy, can anybody acquire at some time a PDF of the ASV program?
That's a good question. I don't know the answer to that.
Or at very least, there must be available a link to certainly the symposium speakers.
Mm-hmm. Sure. So I mean, the program is online, and you could click on. I mean, I don't know that those names of either the plenary session speakers or the state of the art speakers. I don't think those are hyperlinked to their web pages, but you can obviously Google search them. So, in particular, the state of the art speakers, we tend to try and get some up-and-comers, and then the plenary session speakers tend to be those that are maybe a little bit more established, although the lines are blurred, in both directions. So check out the ASV meeting website for some names and some research topics that are up-and-coming, but I would have a hard time limiting myself to any one area of virology that's hot. I just came from study section, and it still seems to run the whole gamut.
Yeah, and I think in particular, entering graduate school, if you enter a good program, the part of their mission should be to expose you to all kinds of stuff, and you just want to try and pick a program where there's a wide variety of things going on, and you'll find out during the first year or so what floats your boat.
Right. Look around at graduate programs. and find places that have multiple virologists, first of all.
Yep.
And the other thing is, obviously look back at our guest list.
Yeah. Right.
Yeah.
We do have one.
And quite a few of our guests have even commented that they have multiple virologists in their departments, and they come from active places in that.
There's a couple of programs that just happen to come to mind with multiple virologists, like University of Michigan, the micro department, right?
Mm-hmm.
Mount Sinai,
University of Pennsylvania.
University of Pennsylvania, University of Florida has a few virologists. You're a little thin in the virology area right now, is that right? You and Steve, right, Vincent?
No, we actually have acquired Ann Moscona.
Oh, excellent!
Oh, that's right. Yeah.
Isn't that great?
So now on
so
this
Columbia,
but Goff and I are old, and so it's not worth coming for us. So yeah, we're thin, I would say, for sure. And stay away. Don't come here. But the places they've mentioned are really good. Also, there are a number of virologists where Julie Pfeiffer is, right?
Yeah, and so that's it.
Where's that?
UT Southwestern,
Southwestern.
Also Madison.
Baylor, yes, Baylor, Madison and Baylor as well. Right.
And we mentioned both Pitt and Penn, is that right?
Penn, not
yet.
Pitt.
But Pitt definitely does.
Yeah,
Yep.
Pitt does.
Addition of a couple in the last year, in fact.
Yeah. But you should also look at the papers we talk about on TWiV, because we tend to pick things that we think are neat and exciting and may often be new areas. So you're listening, so you've got lots of ideas there.
Yeah.
All right, let's do one more. The next one is RIS. Is that how you say that? RHYS? I've recently discovered your channel, and it has been helping me with my virology revision. What is a virology revision? Is that some kind of a test somewhere?
I don't
I don't know.
I have a couple of questions. One, do you have a reference for adenovirus protein 6 destroying the endosomal membrane, thus allowing entry into the cytoplasm? Kathy has put some references there for that, right?
Yep. Chris Wiedhoff's work.
And 2, poliovirus generating the pore-like structure, allowing the genome entry into the cytoplasm. And I've put a couple of papers there for you from Jim Hogle and Marie Chow. Also, and this is why this is the last, what do you think are some of the major similarities and differences between the entry of envelope and non-envelope viruses? Hey, you got to do your own work,
Yeah, I think this virology revision might be
Probably.
an exam
It's
or
over.
Open book.
Homework,
Yeah, we're too late. Yeah.
But
Oh, well.
the major similarity is that the genome has to get out of a particle.
Yes. Major difference is that one is enveloped and the other is non-enveloped.
Yeah,
Right.
different mechanisms, and you've got it right there. The two viruses you're talking about, the virus genome has to get out of a capsid, right? So that's a different problem from a membrane that can fuse.
Mm-hmm.
But of course, some viruses have both a membrane and a capsid, and there you have multiple issues. But those are good questions, and probably the revision is over, but it's okay. Yeah, we have a bunch more left here. My gosh.
Boy, do we.
My gosh. We'll have to do an all... But people don't like all email episodes.
Oh, no.
Really?
Some people have... There's mixed opinions. Some people have complained. Other people like them. And we haven't done one in a long time. So those who don't like it can just suck it up. It's okay.
We were keeping up with the email for a while, and now we've fallen behind. I think we need to... Our next... Well, after we...
Yeah, well, the problem
is late July or something.
When we do live episodes, we don't do email, so it backs up. Which is not to say, folks, that you shouldn't send in email. We'll get to it.
Right.
It's just that when you're talking about 4 degrees weather, it's going to be August.
Yep.
Right. Let's do some picks. Kathy, what do you have?
Oh, what did I end up picking for today? I'm scrolling down.
A lot of scrolling.
Paragraphs.
Oh,
5 pivotal paragraphs in a paper.
5. Oh, I thought you were telling me to look over at the outline links on the left. Yes, the 5 pivotal paragraphs in a paper. This is a blog post that is kind of interesting. It's from Dynamic Ecology, which I follow. It's written by 3 different people. One of them is a woman faculty member here at Michigan. And this starts out, it's a person who is an editor-in-chief for a journal where he has to make quick decisions looking at 600 papers a year as to whether or not to send them out for review. So he gets very good at skimming them. And so he's got what the important 5 paragraphs are. And so if you're in the midst of writing a paper, you might want to take a look at this, because it's just a really pithy analysis of what you need to have in each.
It's actually very interesting. This is because I look at this, and by God, this is kind of the way I skim papers.
Mm-hmm.
Yes.
Yeah.
First paragraph of the introduction, last paragraph of the introduction, first paragraph of the discussion, last paragraph of the discussion, and the abstract.
Yep.
I'll be darned.
Mm-hmm.
That is assuming, of course, that the paper is well written.
Mm-hmm.
Right.
And when you're, as you said, Kathy, if you're writing a paper, you might keep this in mind. And this is a beautiful picture of a fox and some lupin or something.
Yeah, that's always on their blog page. Mm-hmm.
We got a fox living in our backyard.
Oh.
See it all the time. In New Jersey, of all places. Really cool with that big fluffy tail. Neat. Alan, what do you have?
I have a YouTube video and also the associated YouTube channel that I just came across recently. The video I picked from it is called All About That Space. It's a send-up, well, not exactly a send-up, but a reworded cover, a parody of the All About That Bass song that was a big hit a year or two ago. This is a physics parody, and I think it's quite well done. And the rest of the YouTube channel by this fellow goes by the username Coma Nitty. It's raps on various scientific subjects, and I think it's quite good.
Excellent.
All About That Bass. That's right. There was parodies of that previously,
Yes.
right?
Yes, and I think that's when this came out. This is I'm late to the party. Fun channel.
All right. Rich, what do you have?
I'm still on my science fiction kick here. I'm recommending a science fiction series called The Expanse by James S.A. Corey. Now, this actually starts off as a series of books. There are currently, I think, 5 books in the series, and this guy releases about 1 a year, and his target is something like 9. And part of the first book has been made into, a movie series by the Sci-Fi Channel. I think there's about 7 episodes in the series. My son pointed this out to me, and so I binged watched the Sci-Fi Channel series, and then I mentioned it to my bug lady, the woman who comes around and discourages insects from invading our house, who turns out is a sci-fi freak, and she sent me all the ebooks for the books, and I've read the first one, and it's all outstanding. I would actually go for the books first, but the Sci-Fi Channel series is really good. So, great stuff.
You're doing
Cool.
sci-fi, huh?
Yeah, I love sci-fi. It just really gets sucked in, and these are actually quite well written, and the series is well produced. And this is classic. Humanity has occupied the solar system, and we're dealing with stuff from outside the biological, apparently stuff from outside the solar system that's creeping in, and it's all cool.
Independent of your recommendation, my son read Dune in the last couple of weeks, right?
Ah, okay.
Our older son, and he loved it. He just
good.
And the other day, a couple of weeks ago, he goes, I know this is a terrible movie, but I have to watch it. So he sat down to watch this horrible Dune make.
Yes.
He just, every 5 minutes he was saying why it was wrong and it wasn't in the book and why did they do that? It was just so bad. It is.
Yeah.
Why would you make such a bad movie?
Yeah, they really messed it up.
Oh my
It's too
gosh.
bad.
Oh my. And it starts to rain at the end.
Yeah.
What the heck is that all about? It's been so long since I've read it, but anyway, yes. So I have for you kind of a continuation of a pick that I did a couple of weeks ago. The American Society for Microbiology ran its Agar Art Contest this year. I showed you all the entrants a couple of weeks ago. And now we have the winners. First place, fertilization. It's pretty cool. Second place, This Is Not a Beer. Third place, 12 Years of Yuck. And People's Choice, Bacterial Shadow of Wolf. So they're quite nice. They're quite nice. They're very beautiful. They're all very pretty. Mm-hmm. And then we had a panel of judges voting, and then they also took likes on Facebook into account. They got 117 entries from 26 countries and 17 U.S. states. And these will be showcased, by the way, at ASM Microbe next week in Boston, the winners, that is. So that's very, very, very cool stuff, and looking forward to it next year. We have a listener pick. Ted writes, Dear TWiVers, as a listener pick, I suggest turning the pages online for viewing the Marshall-Nirenberg charts. The first summary, this NIH National Library of Medicine project features 10 lab book images and information on the remarkable career of Marshall-Nirenberg, 1927 to 2010, who shared the 1968 Nobel Prize in Physiology or Medicine with Robert Hawley and Har Gobind Khorana for their interpretation of the genetic code and its function in protein synthesis. Especially noteworthy is the included reprint of Nirenberg's Science editorial of 11th August 1967 entitled Will Society Be Prepared?, which contains his prescient concern about the future resultant manipulation of human genes. The concluding paragraph begins, quote, The point which deserves special emphasis is that man may be able to program his own cell with synthetic information long before he will be able to assess adequately the long-term consequences of such alterations, long before he will be able to formulate goals, and long before he can resolve the ethical and moral problems which will be raised. He concludes, I state this problem well in advance of the need to resolve it because decisions concerning the application of this knowledge must ultimately be made by society, and only an informed society can make such decisions wisely. We have obviously entered this debate with the advent of CRISPR-Cas9 and other methods of gene alteration engineering, which you have discussed on past episodes, and of note are the April 11th Wall Street Journal article, Should Heritable Gene Editing Be Used on Humans?, with the pro stance written by George Church, professor of genetics at Harvard Medical School, and the opposing con article written by Marcy Darnofsky, executive director of the Center for Genetics and Society. Additionally, the April 14th, 2016 New England Journal Perspective article entitled The Public and the Gene Editing Revolution by Robert Blendon and al. Attached open access discusses the public's present view of gene editing. Again, many thanks from sunny Florida, present temperature 84F, for continuing to brighten our days with your wonderful podcasts, which continue to provide an excellent window into the scientific literature and knowledge advances. Ted Splaver is a DMD and adjunct faculty at the College of Dental Medicine at Nova Southeastern University in Davie, Florida.
Cool.
Thank you, Ted. Yes.
Excellent. Very nice.
I didn't know that Marshall-Nirenberg could see where this was going. Very cool.
Yeah.
Good to be prescient. That'll do it for TWiV 393. You can find us at iTunes, microbe.tv/TWiV, and also at Google Play Music, and wherever fine and free podcasts are distributed. You can listen to them on your iOS or Android devices with the correct free apps. We do love getting your questions and comments. Please send them to TWiV at microbe.tv. And please, again, consider supporting our work at microbe.tv/contribute. Kathy Spindler is at the University of Michigan in Ann Arbor, Michigan. Thank you, Kathy.
Thanks. This was fun to be back.
Rich Condit is at the University of Florida in Gainesville. Well, he's not really there. He is a emeritus professor. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove is at alandove.com. You can also find him on the Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.